Hello, first of all, let me introduce myself. My name is Yohanes Holanderi. I am a physician nutrition specialist at Faculty of Medicine, Universitas Indonesia. I am delighted to be here today to tell you about the roles of zinc and selenium in metabolism. I'd like to start by talking about zinc. Zinc has many seemingly divergent functions, probably because it is a component of numerous metal enzymes. As a component of metal enzymes, zinc provides structural integrity to the enzyme by binding directly to amino acid residues and thereby stabilizing the enzyme's tertiary structure and participates in the reaction at the catalytic site. According to this report, zinc also plays a major role in regulating gene transcriptions. This leads to my next point, which is role of zinc as acid-base balance. Let me use this figure to show that carbonic anhydride has a very high affinity to zinc, which plays a catalytic role. Carbonic anhydride found primarily in erythrocytes and in renal tubule cells. This enzyme is essential for acid-base balance and respiration. Let's now turn to another role of zinc in alkaline phosphatase. Alkaline phosphatase active site found on the enzyme where 1 magnesium and 2 zinc ions located has the function of acting as a catalyst site. So that means zinc also stimulating osteoblastic bone formation and mineralization by stimulates gene expressions of various proteins including alkaline phosphatase. Okay, next up, role of zinc in alcohol dehydrogenase. If you look at this picture, you will see that alcohol dehydrogenase also contains 4 zinc atoms in enzyme molecule. Alcohol dehydrogenase is important in NADH-dependent conversion of alcohols to aldehydes. For example, alcohol dehydrogenase converts a form of vitamin A, retinol, to retinol, that we know retinol is needed for the visual cycle and night vision. Zinc plays a role in carboxyl peptidases A and B. Carboxyl peptidases A and B are exopeptidases secreted by the pancreas into the duodenum are necessary for protein digestion. The enzymes cleave amino acid from the amino terminal end of proteins or polypeptides that are being digested in intestinal tract. This picture below shows that zinc is bound tightly to carboxyl peptidases. That means zinc is essential for enzymatic activity. According to the literature, enzyme activity decreases with zinc deficiency. Zinc also plays a role in delta-aminolevulinic acid dehydratase. It catalyzes the condensation of 2 delta-aminolevulinic acids to form 4,4-bilinogen that involved in heme synthesis. We have to remember that zinc is essential for the maintenance of free thiols in this enzyme because zinc prevents the oxidation of thiols groups and consequently disulfide bone formation within the enzymes. Superoxide dismutase serve important antioxidant defense roles in the body by catalyzing the removal of superoxide radicals. Superoxide dismutase found in the cell cytosol requires 2 atoms each of zinc and copper for function. Zinc also appears to have structural role in the enzyme. Zinc plays a role in phospholipase C for phospholipid metabolism. Phospholipase C hydrolyzes the glycerophosphate bond in phospholipids. From this figure, we can understand that phospholipase C requires zinc atom for catalytic activity. Okay, next up is role of zinc in polyglutamate hydrolase for folate digestion. As we know, folate is found in foods bound to several glutamic acid residues. For folate to be absorbed, all but one of glutamic acids must be removed. The enzyme polyglutamate hydrolase removes all but one of the glutamic acids from folate. It should be emphasized that polyglutamate hydrolase is a zinc-dependent enzyme that contains zinc atoms. Let's now turn to role of zinc in matrix metalloproteinases for wound repair matrix. The matrix metalloproteinases generally function in wound healing, degrading components of the extracellular matrix to allow for remodeling of extracellular matrix proteins and tissue repair. Matrix metalloproteinases include a group of zinc-containing endopeptidases. It should be emphasized that zinc location is at the catalytic site where the substrate binds. This leads me to my next point, which is a role of zinc in gene expression as a major role in regulating gene transcription. To perform this function, zinc binds to transcription factors called zinc fingers. Zinc fingers interact with specific DNA sequences called metal response or regulatory elements that located in the promoter region of selected genes to either enhance or repress transcription. Now I'd like to move on to the last part of zinc role in insulin and carbohydrate metabolism. Zinc is involved with insulin and thus influences carbohydrate metabolism. Zinc is transported into pancreatic beta cells by zinc transporter zinc T5 and zinc T8. We know that pancreatic beta cells are responsible for insulin production and secretion. That insulin is stored with zinc in granules in the pancreatic beta cells until it is released into the blood. Zinc also appears to regulate the protein kinase mammalian target of rapamycin or mTOR, thereby affecting insulin signaling and protein synthesis pathways that occur when insulin binds to cell receptors. Zinc is widely found in tuna, egg, meat, and also chicken, and also plant protein as such as mushroom. 13 milligrams a day and 10 milligrams a day is needed, respectively, in men and women. There's a lot more to learn about the role of zinc, but since we're pushed for time, let's move on to the role of selenium. Various defined roles of selenium include its involvement in maintaining and inducing the cytochrome P450 system, in DNA repair and enzyme activation, and in immune system function. Moreover, roles of selenium are related to its function as an integral part of specific enzymes in the body that called selenoproteins. Selenoproteins have primary function in antioxidant capacities and thus regulate cell redox status. Selenium plays role as integral part of glutathione peroxidase. Glutathione peroxidase catalyzes the removal of hydrogen peroxides and organic hydroperoxides. If hydrogen peroxides not removed, these peroxides typically damage cellular membranes and other cell components including proteins and DNA. Another role of selenium is as the major part of selenoprotein. Selenoprotein P is thought to function in the body as an antioxidant, especially in removing the damaging peroxynitrite radical. If peroxynitrite not removed, can cause DNA single-strand breaks and lipid peroxidation. Selenium is also necessary for iodine metabolism and may regulate in thyroid hormone production. The thyroid gland contains several selenoproteins including selenoprotein B, glutathione peroxidase, thyroreductin reductase, and also iodothyronine 5-deiodinase. The latter are selenocysteine containing enzymes with the selenocysteine present at the active site. Deiodinases type 1 and 2 to convert the T4 to T3. That we know T3 is the body's primary hormonal regulator of metabolism as well as of normal growth and development. Several other selenoproteins containing at least one selenocysteine have been identified, but little is known about their functions. Like selenoprotein W and M play antioxidant roles, selenoproteins S and also K appear to have roles in the processing and or folding of proteins. Selenoprotein S is to remove misfolded proteins as well as to protect against oxidative damage. Selenoprotein H, found in the nucleus, may act as a transcription factor to upregulate the expressions of the other selenoproteins. diets. Food sources of selenium, especially from animal protein such as tuna, beef, chicken, shrimp, and oyster, and also plant proteins such as shiitake mushrooms and tofu. Daily requirement of selenium are 22 to 36 nanograms a day, and 19 to 26 nanograms a day is needed, respectively, in men and women. Well, that's just about the roles of zinc and selenium in metabolism. Thank you for being here today and listen to my presentation. I hope you have learned something about this section.